Welcome to Datta Biology class again. So we are in the next episode of respiration. In the first episode of respiration, we have completed the glycolysis part. But a little bit of the glycolysis was left over. I will start right from them. Let us directly jump into the subject first. So, gentlemen, you have seen there are 6. 1, 2, 3, 4, 5, 6. Actually, it is 6 carbon compound. It is known as glucose. As you know, we have completed earlier. So, this glucose during glycolysis was pulled apart. Pulled apart to this 3 has gone to some side and this 3 goes to some other side. So, this is, we know that it is phosphoglyceraldehyde PGALD 3-phosphate and one is dihydroxyacetone phosphate. These are all 3 carbon each. Now further it has come to pyruvic acid. So this is pyruvic acid. Formula is pyruvic acid CH2 COOH COOH and then CH2 COOH. So this pyruvic acid has got 3 carbon atom in its molecule. Anyway, that is not a part of discussion. Now these 3 pyruvic acid we have produced in number 2. That is, it has been converted to this one again. I told you on the other day that dihydroxyacetone phosphate can be converted to phosphoglyceraldehyde with the enzyme known as triose phosphate isomerase. So there are 2 PGLD. Those 2 PGLD has been actually produced 2 pyruvic acid. It is a 3 carbon compound. Now up to this is known as glycolysis. This is known as glycolysis. Feel free to go to my earlier class where I have written, it was a little bit lengthy, where I have already discussed with the glycolysis part. Now, why glycolysis? Why this? So this will go to Kreb cycle. I told you on the other day, and as you know, that this will go to Kreb cycle. So before entering into Kreb cycle, this pyruvic acid is not capable of entering into the Kreb cycle or in mitochondria where the Kreb cycle takes place. So this 3 carbon compound has to be produced as 2 carbon compound before it enters into the mitochondria. Why? Because let me say, suppose this is mitochondrial wall, outer membrane of the mitochondria. Okay. Now this inner membrane is infolded. Inner membrane is infolded to form cristae. As you know, on the other cellular organisms we have already come across. This 2 carbon COA is a good transporter of carbon because it is only, say for example, let me start here. It is a small opening. So from this small opening, this 2 carbon can enter. So in order to enter and to complete the cycle, this requirement is necessary. Now this glycolysis has produced this to this. Now from this to this, it is a link reaction because this is a linkage, and until and unless you form it, you cannot enter. That's good. Now in glycolysis, why we are doing all these things? Because we want to produce ATP, the energy currency. What we have produced here itself is 2 ATP and 2 NADH plus H plus. So these 2 are the products we have completed, prepared in the glycolysis part. Because This ATP is a direct currency node, and these two NADP, from these two NADP through a chain, electron transport chain, will produce 3 ATP. So that is the indirect way of production of energy currency. This is the direct way. So direct energy, indirect energy production. This we have produced. Here what we will do, this we have to produce this. Now what we have to do? We have to insert this. So there is an insert of CoA, coenzyme. Likewise, you see there are 3 carbons and here it is 2 carbons. So 1 carbon must go. So 1 carbon should go in the form of CO2. So it is a case of decarboxylation, isn't it? Then let me say that coenzyme will enter here and CO2 will go out. Am I visible? Okay. Now this coenzyme, what you will do? Coenzyme, during this, some NAD is converted to or reduced to NADH plus H plus. This is NAD is taking hydrogen, so NAD is reduced and it is giving out hydrogen, it is oxidized. So this is a case of oxidation and this is decarboxylation. So somebody may say that oxidative decarboxylation of pyruvic acid produces acetyl coenzyme. This is somewhat known as ACETYL, acetyl CoA. Okay. So this is dehydrogenation. So in fact, here one NADH2 is produced. Why 1? Because there are 2 pyruvic acid, so it should be 2x. 2 pyruvic acid. One thing I will say that because of converting pyruvic acid to acetyl coenzyme, there is a chain of reactions. In the chain of reaction, 2 enzymes are required. Number 1 is TPP. TPP, otherwise known as thiamine pyrophosphate. And another enzyme is known as lipoic acid. LIPOIC, lipoic acid. These are the 2 enzymes required for the conversion of pyruvic acid to acetyl coenzyme. How does it act? Pyruvic acid mixed with thiamine pyrophosphate, it gets attached here with the thiamine pyrophosphate. It gets attached and it produces an active aldehyde. And from this active aldehyde, 1 carbon, after losing 1 carbon, 2 carbon is there. These 2 carbon gets activated. So it is known as activated aldehyde. Then it reacts with lipoic acid and aldehyde group enters. So there are the possibilities of 2 enzymes in this reaction. The one thing somebody may not say, but I want to say that, this is thiamine pyrophosphate. Thiamine, what do you know? Thiamine, THIAMINE. Thiamine is vitamin B1. Vitamin B1 is otherwise known as thiamine. Here coenzyme is somewhat related to pantothenic acid. PANTOTHENIC, pantothenic acid. This is vitamin B3. Here you have seen niacin, somewhat related to niacin. Niacin is related to NiACIN, vitamin B5. So vitamin B1 is required, vitamin B3 is required, vitamin B5 is required. These are vitamin B complex, as we say. Vitamin B1 is known as thiamine, vitamin B2 is known as riboflavin, and vitamin B3 is known as, here I have written, pantothenic acid. Then vitamin B6 is pyridoxine, vitamin B12 is cyanocobalamin. These are the names as we know, all these things. So what we have seen that there are 3 vitamins required for the conversion of these. Other than vitamins, no glycolysis and no Kreb cycle can take place because of these vitamin. So doctors used to give vitamin along with, because to increase the mitochondrial cycle, to increase the rate of energy, so vitamin B group is required. So here what happens? Once it is produced, it is a good packers and movers. So with the help with the car, they are taking these 2 carbon inside the mitochondria. So within the mitochondria, that something will go out. Anyway, what happens? In the mitochondria, it enters these 2 carbon. So while entering, this CO is released. CoA, coenzyme is released, because once it is entered through this narrow path, then this track is not required, only the carbon is required. So this carbon, along with this carbon, this is already present within the inside the mitochondria. It is known as oxaloacetic acid, or somebody says oxalate. So this oxaloacetic acid is present here, and it takes 2 carbon molecule, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. It is citric acid, CITRIC, citric acid. Citric acid produced here. So one thing I have not said, that here the enzyme is known as pyruvic. From pyruvic I am taking the name, and this is hydrogen is going out. It is known as pyruvic dehydrogenase. Pyruvic dehydrogenase. Write it. Pyruvic dehydrogenase. Now another enzyme. Here it enters, and this oxaloacetic acid and this one is required. So it is producing citric acid. It is known as citric synthetase. What is known as citric synthetase? SYNTHATASE. Citric synthetase is the enzyme which converts this oxaloacetic acid already present in the mitochondria. It mixes with 2 carbon and produces citric acid. Citric acid, the structure of citric acid is somewhat like that: CH2COOH, CCOOH, and CH2COOH. These 3 are known as carboxyl group. OH is hydroxyl group, double bond CO is acid, some other ketone group. So these 3 carboxylic acid group is present. So this cycle is known as tricarboxylic acid or TCA cycle. And since citric acid is first produced, it is known as citric acid cycle. And it has been discovered by Kreb, hence Kreb. It is known as Kreb cycle. It is known as Kreb cycle. And what we know more, that is this cycle runs within the mitochondria. So we should name as mitochondrial cycle. Mitochondrial cycle. So these are the 4 names: TCA cycle, citric acid, Kreb cycle, and mitochondrial cycle. These are the name of the cycle. This is not that much important, but once you can remember, it can be remembered easily. Now let us start with another one. This citric acid is converted to isocitrate. This citric acid is converted to isocitrate. Here I have already written COA. Anyway, so this is the 6 carbon, this is the 6 carbon, they are actually isomer. So isocitric, and this one is citric acid, this is mediated by aconitis. The enzyme is known as aconitis. With the help of aconitis, some isocitrate, they are all isomers. So in some books we will come across the citric acid, it goes to, it becomes cis aconitic acid, then isocitric acid. In between there is a step wherein some H2O is entered and H2O goes out. So hydrogenation, dehydrogenation takes place. That is not much important. With the help of aconitis, isocitrate is produced. This is all 1, 2, 3, 4, 5, 6. So this 6, this 6, this is 6. This is also 6. But it is not glucose, it is citric acid. Now, gentlemen, from isocitrate, what you see here, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. So, easily you can say this, that losing CO2. CO2 will go out. And here also, NAD will be converted to NADH plus H plus. Actually, decarboxylation takes place and oxidation of take place. So there should be an enzyme in between. This is known as isocitric dehydrogenase. It's known as isocitric. This is dehydrogenation is going on. ISO-CIT RIC dehydrogenase DHYDROGENASE isocitric dehydrogenase within the mitochondria it takes place. What I forgot to tell you that I will come later on. Within the mitochondria, these are enfolded inner membrane. So here there are so many liquid is this liquid is there is outer side and inner side matrix is there, and here some table tennis rackets are there. These are known as oxysomes. OXYSOMES, oxysomes. Here ATP synthesis takes place. We'll come later on. These has got one head piece, one stop, and one is base piece. Head piece is known as F1 particles, and stop and base piece collectively known as F0 particles. So within these particles we will come later on. These are called oxygens, which I forgot. I may forget in future, so I am telling all these things. Now some CO2 is going out and NADH is produced. It is called isocitric dehydrogenase, and this is produced alpha keto glutarate. It is known as alpha keto glutarate. This was the alpha keto glutarate or glutaric acid, but we used to say alpha keto glutarate. It is a 5 carbon compound. Gentlemen, you see this 1, 2, 3, 4, 5, 1, 2, 3, 4. So another carbon is losing, is it not? So here another carbon is losing. So CO2 is coming out, decarboxylation. What you see? The same thing what we have seen here itself. COA is entering and this CO2 is going out. So some NADH will be there. So here NAD will be converted to NADH plus H plus. In fact, this is a case of dehydrogenation. So name the compound, name the enzyme as alpha keto dehydrogenase. Alpha keto known as alpha keto dehydrogenase. Enzyme converted this. This is known as coenzyme. This is actually known as succinyl coenzyme. SUCCI. Succinyl coenzyme. Here coenzyme have not entered here anyway. Some COA will come in. The same reaction we have come across here. This is with the pyruvic dehydrogenase. Here alpha keto dehydrogenase. Here CO2 going out. Here some COA is entering. So here also you see this is vitamin niacin. Niacin is this one. And this is pantothenic acid. Vitamin B3. Here vitamin B5. So some vitamin is required within the mitochondrial cycle. Doctor used to give vitamin along with your patient for increase in mitochondrial respiration or respiratory increase in the respiration. So this cycle can go and it will produce more and more energy or ATP currency, directly or indirectly. Because it has indirectly here it is produced. Now no ATP has so far been produced. Succinyl coenzyme is converted to succinate. Will be converted to a succinic acid, SUCCI, succinate, NAT. This will be converted to succinate. This is also 4, this is 4. So CoA will go out. Here there is no CoA. Simply write in that way. CoA will go out. Coenzyme will enter here. This will perform the and it will track will go after delivery of the products. Here succinic acid is produced, is known as succinate synthetase. Succinic synthetase, with the help of succinic synthetase enzyme, coenzyme is removed and succinic acid is produced or succinate is produced. Here one more thing is produced. I have given here that no ATP has been produced. Here ATP will come. Let me say here one GDO. Guanosine diphosphate will be converted to guanosine triphosphate. GDP to GDP is one kind of ATP. The currency in other form. So here one inorganic phosphate is required. Inorganic phosphate IP is required. GDP will be immediately converted to GDP and this phosphate will be taken over by ADP and will be converted to ATP. So here indirect way, it is direct way. It is producing in direct way. Now let me see this succinic acid, oxygens are just taking out. Succinate fumaric, malic and this. Fumaric acid. Succinate will produce fumaric acid or fumarate, Fumerate, fumarate. Here NAD here, FAD, flavin adenine dinucleotide, flavin, riboflavin, adenine dinucleotide in dinucleotide, adenine dinucleotide you have gone through in DNA, deoxyadenylic acid, adenine dinucleotide, there are two nucleotides, flavin adenine dinucleotide will be converted to FADH2. So here FADH2 will be produced, and succinate to fumaric acid will be done by succinate dehydrogenase, because hydrogen is going out. This succinate dehydrogenase. Succinic acid will be converted to fumarate with the help of enzyme succinate dehydrogenase, and here FAD will be produced. Yeah. Okay. Will produce malate, MALATE, or malic acid. Fumaric acid to malic acid or fumarate to malate. Here a little bit water is required. H2. So from fumarate to malate, enzyme fumarate. Fumarase enzyme helps in conversion of fumaric acid to malic acid. Malic acid, again, malic acid, here 4,4, nothing, no problem. 4,4, no problem. Here, again, NAD will be converted to NAD. NAD will be converted to NADH plus H plus from malic, conversion of malic to oxaloacetic acid, OAA, or simple oxalate. So this is a case of dehydrogenation. This is a case of dehydrogenation. Another, this is a case of dehydrogenation. So let me say it, malic dehydrogenase. Malic dehydrogenase. MALIC, malic, DHYDROGENASE. This is a case of malic dehydrogenase is the enzyme which dehydrogenate. Out of dehydrogenation, the NAD is reduced to NADH2. Now the cycle goes on. The cycle goes on in this way. Now what we did earlier, here one acetylcholine we have taken into consideration. Actually there are two acetylcholine because one pyruvic acid produces this and two pyruvic acid will produce two. So everywhere there will be two. So here you see NADH is produced 2. NADH is produced 2. 2 to 4. Here itself 2. 6 NADH is produced here. 6 NADH. Let me say NADH2. Okay, for our currency purpose. And here FAD is produced. the smaller brother of NAD, the younger brother. So FADH2, 2, it is molecular 2, that is 2 FADH2 will be produced. And how many ATPs are there? You see, here no ATP, no ATP, no. Here 2 ATP is produced. 2. Because so 2 ATP is produced. 2 ATP is produced. Clear? Not this much. Let me say. Anyway, I'm erasing this one, okay? What we need actually 2 ATP. Here 2 ATP. 2 NADH2. Here this NADH2 has not taken care of. These 2 NADH2 make it 4 NADH2. 4 NADH2, 1 NADH2 will produce 3 ATP. The 3 4 are 12. It is 14 ATP is produced. Currency. 14 currency note has been produced. Here 6 NADH2 is 3 6 18. 18 ATP. 2 FADH2 each at the rate of 2. It at the rate of 3. 3 6 18. 2 2 4. 2 ATP. It will produce 24. Adding grand total 448 213. 38 molecules. 38 molecules of ATP are produced. And one thing more I like to say that lipid and fat. Here, this acetyl coenzyme can be produced from lipid and lipid is fat, lipid and protein. This is the extra point you can write or you can keep it in your mind that lipid and proteins. So protein, glucose is carbohydrate that we have gone through, protein and lipid on degradation, on D, write it down, GRADATION. On degradation produces acetyl coenzyme. So acetyl coenzyme is the key compound to our knowledge. Pyruvic acid is all the key compound, but for the production of ATP, protein from lipid, this fat, protein, carbohydrate, protein, fat, there is a linkage. And this pyruvic acid can be produced by in the cardiac muscle, and in the liver with the help of lactic dehydrogenase. This can be converted to, this is known as dehydrogenation. This is known as here I have seen lactic acid. On dehydrogenation produces pyruvic acid. So it follows the same path. So this is very much important. That is glycolysis in pyruvic acid is important, and acetyl coenzyme is much more important than this. Was what we had in the next stage. We'll pass on the electrons. Whatever electron has been produced indirect way, these are all electron carrier. That is, electron will be passed on to carrier to carrier to carrier. That will take 5 minutes. Not that much lengthy. And this much we know. Thank you very much.